Okay, welcome everyone, and thank you for that introduction. So we've been hearing a lot about the connectivity between organisations. We've been talking a lot about connectivity to offshore, and so our job here is really to talk also about the connectivity between the work that we do and the growers, and the important space they play in that whole biosecurity journey, to make sure that we don't spend all of our time looking out and we also look in, because we know that some of our next problems could actually be turning up under our nose without our knowledge if we're just not looking. So today we're going to talk about our unusual symptoms process, and also we're going to link that to how some grower reports have led on to some research, and how that research has now double backed around to answer some questions on how we can keep our orchards resilient into the future, and just raise awareness, which of course is a key part of biosecurity. So, what do we mean by unusual? Basically, it's anything that appears new, or it's something that has no obvious explanation. It might be a new insect you haven't noticed before. It might be something that looks just a little bit too much like the BMSB, so best go tell someone. It could be out in the orchard. You might see a sudden collapse of a single vine, or even a whole group of vines, and wonder what is behind that. Or it just could be a slow, poor vine health problem that's occurring on your orchards. So bringing those unusual symptoms together from across the industry, and remember we are across a number of regions, so there will be regional effects, just allows us to capture, consolidate, and also provide diagnostics for the things that are reported out there on the coalface. So this is the process that we use. It's pretty straightforward. People get in touch with KVH either directly by phone call, through our email system, or we've also got an online form through which you can report to us. When it comes in terms of pests, we refer to industry partners, specifically Plant and Food, to make sure that we're making the right call on the things that we're seeing. And if that's beyond our knowledge, it goes on up to the Plant Health and Environmental Lab in Auckland to verify what pest has been found there. In terms of the unusual vine symptoms, first of all, we have to think about, is what we're seeing due to a nutritional or an environmental or perhaps a management-related cause? And again, we go out to our industry technical groups to help with that. And then we go out to the orchard and we take samples, and they move through to our diagnostic labs. So the go-to for a lot of that work is plant diagnostics in Christchurch, and because it's across there's a need for KVH to do a movement permission for that, because that's part of our pathway plan requirement. So we work through that. And then beyond that, the results either flow back directly to the growers, or if there's anything new found at any step along the way, they all move to the Plant Health and Environmental Lab to verify that yes, it is new, and then the MPI spaces to decide whether there might need to be an investigation. If there is an investigation needed, KVH can work in alongside MPI and do the steps that they'd like us to be involved in, and we also look to take that grower along that journey, so they're just not left out in never land while that's going on. Across the years, we've built some very strong relationships with our labs and partners, and I can say that Plant and Food in particular is fantastic at being there on the Friday night at 8 o'clock when you get a call, or on the long weekends, which is generally when biosecurity incursions occur. So what sort of reporting have we been getting? So on the left-hand side there, you've got a bit of a look at the reports that came through for our 23-24 reporting year. So 48 vine symptoms coming in from the industry, and 32 pests. There was far more beyond that, because a lot of them get dealt with quite quickly, because the common shield bug quite often gets reported, and we're quite clear on what that one is. Further down, you'll see that the Fowl Lab was involved in working with us on 27 occasions this year. Plant and Food assisted help for 26 of our cases. 5 samples went down to plant diagnostics, and then the PSA ones go through the Hills Lab, unless it's a PSA related sample from the South Island, so that gets straight up into the FEL lab to make sure that we haven't got PSA down in that space. There was also 2 investigations of new to New Zealand things. First of all was a eucalyptus eating beetle, probably from Australia we would suspect. And the investigation in that case identified there was no additional biosecurity risk for the kiwifruit industry or other sectors, so that one was able to be closed. And the other one was regarding a Phytopithium helicoides species, so that was something that had not previously been found associated with kiwifruit. Fortunately, we had some samples archived from previous research projects, which helped to show that this indeed was not a new biosecurity incursion for New Zealand. So both of those 2 investigations were able to be closed off, but that's just showing how the system is working, and no calamities have occurred. This is just a quick look at the number of organisms that have been pulled out of the unusual symptoms that the growers have we've worked through with the growers. And there's 6 new associations with kiwifruit within that, so you can see we keep building some knowledge there. But what we want to do today now is to share how some of the early reports of unusual symptoms have worked their way through to a research project. So at this point I'll hand over to Joy to describe that research journey.
Thank you. So this is just really a timeline of the unusual symptoms of trunk diseases over time. It's not new. It may look like it's new. We're getting all of these new symptoms coming through, but it really started back in 1995 when reports from Italy were coming in of these unusual woody symptoms on kiwifruit. By the time we hit 2000, those woody symptoms were starting to be seen in New Zealand, so we were seeing them on Hayward, and then very quickly in Hort 16A. And at that time, the pathogens that they were finding associated with those were Neobulgaria alba, cylindrocarpin species, and verticillium species. At the same time, we were still getting reports of pathogens of kiwifruit wood from overseas, but they're starting to include members of the Nectriaceae fungal group. So Ilionectrias, Fusarium solanae, other Fusaria were coming through. Kept on getting hints that there was something wrong, and then in 2010 PSA was discovered in New Zealand, and of course everyone dropped everything to deal with that. By 2012 New Zealand was really beginning to graft to those new cultivars, but by 2017 you're starting to get those unusual woody symptoms reports coming back in again, and so that was in Hayward again, but it's also the new cultivars like Gold 3. So many of those were only 5 years old, so that's very quickly they were starting to be seen. So over time, these unusual woody symptoms have been referred to as many different things, but we're currently referring to them as kiwifruit trunk disease, or KTD for short, basically to capture all of those diseases of the woody parts of vines. And the symptoms that we're seeing, they're really along a spectrum, and they go from the ground all the way up to the ends of the leaders.
Okay, so the first case study that came in was a report made to KVH in 2017. So that was from a Te Puke orchard, quite a 30-year-old plus orchard, and that had been grafted across to Gold 3 in 2012. It was on Bruno rootstock, and there was declining vine health in the orchard. The grower had already removed about 30 vines, but there was also additional vines on that orchard that weren't looking well. So that led to the beginnings of, or added to our Nectriaceae research projects.
Okay, so as Linda said, the trigger for this was really those increasing unusual symptoms reports, and of course more Nectriaceae are being found from these symptoms. So along with that, there was still more reports from overseas of Nectriaceae associated with symptoms over there. The Nectriaceae, it's a very large fungal family, and it's got a very large host range, but there's a lot of pathogens within that group, and there's a lot of pathogens of woody plants. So the questions around this were really, what organisms were actually being found here associated with that vine decline. How big is the problem? How many vines are we actually seeing that have this problem? So we went out and we surveyed 3 orchards. 2 of these were in the Bay of Plenty, 1 of these was in Motueka. And we did wood coring at different heights on the vine. So basically we get a little core like at the bottom there. And you can see what is actually growing across the vine, and doing it at different heights on the vine means we can see what's happening up and down the vine as well. And so we went and identified the organisms in 10 symptomatic and 10 non-symptomatic vines in each of these orchards. So very, very high level here. We found an astounding amount of symptoms out there. Anywhere between 18 to 34% of the vines in these 3 orchards were visibly diseased. So that's when you're actually starting to see the symptoms. That's not when they're getting infected. That's by the time they've developed symptoms. And this figure didn't include the fact that a lot of vines had already been pulled out. So some of the healthy vines were actually just little striplings that had just been planted. So we found there were many organisms within the woody tissues in those diseased vines. And we found there was quite a difference between the orchards in those symptomatic vines. So orchard 1, which was a Hayward in Te Puke, the main group in there was the Ilionectria species group, with the main species being Ilionectria robusta. We're also finding Neobulgaria alba, which is not within the Nectriaceae, but we were finding that it was quite prevalent, so we did note it. Orchard 2 was a gold 3, again in Te Puke, and the main thing that we found there by quite a significant amount was the Fusarium solani species complex. Down in Motueka, in the gold 3 down there, we were starting to see Neonectria microconidia. This is the Neonectria that Rebecca was just talking about, forming quite distinctive symptoms. Cankers in the trunk, cankers in the leaders. And down there we were also seeing quite a lot of Ilionectria as well, but at this point it was actually Ilionectria liriodendri rather than the robusta that we were seeing further north. So this is probably when we discovered that these woody symptoms were really being caused by a complex of fungi rather than just one thing, and that similar symptoms can be caused by different organisms in different places, and also by more than one organism in one place as well.
Okay, we had another report coming in from Te Puke, and this one was in 2019. In this case, we had the typical orchardist moving his old Hayward vines across to Gold 3, and when the manager cut through the trunks, and particularly the trunks of the vines that looked like they had a disorder, that gave us an internal view of what was going on in those vines, and it didn't look good at all. So that led to a second research project to identify what fungi were causing this staining within the vines.
So coincidentally, this one was actually on the same orchard as Orchard 1 in the previous project. So the trigger was obviously those Hayward trunks with the big staining inside, and the fact that they were associated with those less vigorous trunk disorder vines. So the questions here were what organisms were associated with that internal discoloration, and we wanted to add to the knowledge on KTD of kiwi fruit. So again, fungal isolations were made across the trunk, and we kept an eye on whether they were from symptomatic or asymptomatic tissues. And this time it was not solely focused on the Nectriaceae. So again, many organisms found in those woody tissues, but the most common in the discoloured wood on that orchard was that Neobulgaria alba again. And then Ilionectria species group came in next, and then we're seeing Neonectria microconidia up in the Bay of Plenty as well. The most common in the clearwood was actually Chaetomium species, which is just an endophyte, but there was a big difference between what was discoloured, what was coming out of the discoloured wood, and what was coming out of the clear wood. Neobulgaria alba has actually been previously found in the New Zealand native estate, and it has been noted before as being found in swollen trunks of kiwi fruit. So it's a possibility that this is actually an organism that has done a host jump from the native estate and quite likes being in kiwi fruit.
This is the sample. This is the grower report that prompted the Neonectria project that occurred. This was a grower who had been grafting across his Hort 16A through to the Gold 3, and then just a few years after he had grafted, started noting that he was having declining symptoms. But the brilliant breakthrough that he did is he actually did some counting, or with the help of AgFirst down there, did some counting, and in fact found that he had 11% of his real estate was had cankers and severe dieback. So that's what prompted the balloon to go up around research around Neonectria, particularly down south, to see what was going on down there.
By the time we got down there, it was a lot more than 11%. So while the trigger for this project was actually that Motueka report in 2018, Neonectria had actually been found in Kerikeri in 2015 when it had been called into MPI at that point, and they had given it the name Neonectria microconidia, but it was actually a new name at the time, so we didn't know whether it had been here previously and just called something else, or whether it was actually new to New Zealand. We very quickly found some isolates from Te Puke that had been collected in 2013, but we were seeing this big symptom difference between the regions and the cultivars. So the question here was a bit different from the other ones where we had these symptoms and we wanted to know what was actually associated with those symptoms. Here we're looking to see, we know what we've got, but we have questions around that, obviously. So the questions really are, was it new to New Zealand? Is it a new incursion? Where is it found geographically in New Zealand? And why are we suddenly seeing those symptoms? So we went out and we tested old cultures from the culture collections that are under potentially older names, things that we thought may have been called something else in the past. There was a South Island survey where they looked to see how widespread it was down in the Motueka general region. We collated all the records that we found from other regions, and then we went and we pathogenicity tested 4 different kiwi fruit cultivars. And then, of course, there was the North Island Neonectria symptoms survey that Rebecca was referring to before. So again, really high-level results. We found that it had actually been present in New Zealand kiwi fruit for over 25 years. It's found in all kiwi fruit regions in New Zealand, and it's actually widespread within those regions. It actually causes fruit rots, and it has been noted as causing fruit rots ever since Hort 16A was first being grafted on. And of course it causes those cankers on the vines, but it can be asymptomatic in the vines as well. When we did the pathogenicity testing, we found that those Gold 3 cultivar was significantly more susceptible to this than Hayward, Red and Arguta. So this really showed us that it wasn't a new incursion, but it was a new cultivar that was triggering these new symptoms because it was so much more susceptible to something that we already had. I think it's you.
You think it's me?
No, it's me. Whoops. Okay, so why are we actually seeing these new and unusual symptoms? New situations can change the status of plant diseases, and as you've just seen, putting in a new cultivar of unknown susceptibility to something that we wouldn't have even imagined was going to cause a problem, that can really change the situation on the ground. We don't know what new rootstocks are doing. We do know that different growing practices can change the status of things, so just for an example, girdling puts more wounds in, allows more entry places to the plants. Stressed or ageing vines will react differently as well. And of course, when you've got an ageing orchard, you've also got more and more inoculum building up there. KTD symptoms, they actually increase as the vine's age as well because it's a very slow burn kind of disease. It takes a long time from when it gets infected to when you're actually going to see a symptom. Environmental factors will change how diseases react. Frost, extended wet periods, And as you've heard from several people already, climate change. And of course, the move of kiwi fruit growing into marginal growing areas is increasing that stress on the vines. So what else have we learnt? Fungal species behave differently. When it comes to KTD, the different species, the different genera are found at different places on the vines, and they move in different ways. So we've got organisms that move from the canopy downwards, we've got organisms that move from the root or the crown upwards, and we've got a whole load of them that are likely to enter through those wounds, whether they are your pruning wounds, your grafting wounds, your girdling wounds, or the wounds that you're causing as you accidentally hit the bottom of the vine at some point. So you really need to know the organisms that are actually involved to know how to intervene and how they're reacting, what their behaviour is, to know where you're supposed to be pinpointing these. And just as an example, Eleonectria, soil-borne, waterborne, airborne, but it is more prevalent lower down the vine and in the roots. Whereas Neonectria, it's waterborne and airborne, it's not found in the soil, and it's more prevalent up in the vine, up in the leaders.
Okay, so comes back to the grower, what can I do when you just tell me that this is all very hard? One of the best things that we'd recommend is that growers do actually go out and do a bit of a monitor of their orchard, particularly if they're on old rootstock, because something could be building up in there. And quite often, from my experience of going out and visiting growers, they'll say, yeah, I've got a couple of crook vines. We start counting them, and inevitably it seems that around about 10% of vines with an infection is kind of the trigger point when it hits our radar. So if it's a fast-moving thing, that's just a little bit too slow for us to be able to respond. So if we've got the numbers, we can already start making a replacement strategy to make sure that we protecting our productivity base of our orchard, because we know seasonal effects rock us around a bit, but if we keep that good strong base within our sites, we're going to be in a much better place. Hygiene is number 1, 2 and 3. Basically, wounds provide entry points and tools can transfer disease. The rules we learnt through PSA, they still hold tight against fungal diseases. So we've got the wound protection, we've got the hygiene, we've got the cut out the infection, and more importantly, this time round, we need to get that infection out of the orchard because there's a whole lot of inoculum, and that inoculum can just be moving into the orchard depending on the weather conditions that suit it. Really important to get diagnostics done because then we know what we've got in our midst and how much time we've got to act. So at the end of the day, building resilience in our orchard, it is a team game, and at the front row of that team game is actually our growing communities out there. So we talked about it earlier, if we get complacency in that space, we might end up getting ourselves in a whole lot of trouble again. So we just want to keep people recognising the value of reporting those unusual symptoms so we can start seeing trends, the importance of keeping that research going because a lot of these things are quite slow burn, and then the importance of basically getting the diagnostics done, and we've got some great tools and great networks in this space. So we would very much like to thank all our diagnostic labs, Plant Diagnostics, Hills and the MPI team. We'd also like to thank the MPI Incursion team for their good fantastic guidance when we are following up something through the investigation process. We'd also like to acknowledge and thank Zespri for their funding and support of these biosecurity projects, and Plant and Food for their excellent project work and sharing of their expertise. But most of all, we want to thank the technical people, the orchardists, the orchard managers who take that time to report, because if that flag isn't raised, we can't do anything to help ourselves. So thank you all.
Thank you very much, Joy and Linda. Is there any quick question? Yes, hand up. I know time is short. One hand up.
Sorry you had to run. Great presentation, Joy and Linda, as usual. Two-part question. From growers' point of view, how easy it is to ID some of these diseases, assuming these are soil-borne, or, yeah, I did acknowledge the fact that what are the reasons to that these diseases can spread, the inoculum levels. The second part is any indications on the control measures to decrease the inoculum, maybe from the point of view that when we attended the International Kiwifruit Symposium last year, some of the things that were discussed that they could actually address was quite easily through addressing drainage, for example, or aerating the soils or decreasing the fertigation. And thank you.
I've got a short memory, mate. I can't remember that whole long question. Okay, so I guess, as you know, Pranoi and Joy spoke about, we've got some diseases that we do know that they are more canopy-based, like neonectrias and botchospherias. And we have seen from both the research projects and previous work that's been done that you can actually start cutting back through that canopy to see if you've got infection and staining in that wood underneath. And for those types of challenges, you can cut out well beyond that last point where you see the staining. So that provides the opportunity to kind of refresh that vine before letting that infection take over completely. And then from the ones bottom up, unfortunately, they're more troublesome. And then the thing with those is to really get a replacement strategy in place as quick as you can. And there's good ways of doing that. We know that we can string vines from the other side of the bay so that if we decide to make that cut, we don't have to have that dip in productivity, which is always the reason that growers never want to work with things. But without that diagnostics, it's really hard to help growers to make those decisions. And I've forgotten the other part of your question.
What about getting rid of the inoculum?
Oh, the inoculum.
How to keep the inoculum down? Cut out as much dead wood as possible, cut out as much symptomatic wood. Sorry. Cut out as much symptomatic wood as possible, cut out as much dead wood as possible, even if it's not symptomatic. Get rid of it. Don't leave it on the orchard floor. Get rid of all of that kind of debris. All of those little parathecia that you see, little red parathecia that you see in the orchard, you'll see some of them at the table later. Each one of those is a little spore factory. It's not one spore by itself, it's a little spore factory, and it'll be sitting there spitting out spores. Even if you're leaving the dead wood on the orchard floor, it needs to go.